Welcome to IMAT Tutor. Today, we're diving deep into Lecture 9 of Human Physiology, focusing on respiration, gas exchange, and control of breathing. Our goal is to take you from breath to blood, understanding the complete respiratory system at examiner depth. Whether you are aiming for a perfect score or trying to master the core medical principles, this overview lays out the exact roadmap of everything you need to conquer. We will kick things off by exploring the anatomy of the respiratory system, tracing the entire pathway from the nasal cavity all the way down to the microscopic alveoli. Understanding this structural framework is essential because it sets the stage for physical mechanics of breathing. We will analyze exactly how the body alternates between active respiration and passive expiration, paying close attention to the pressure gradient and muscle movement involved. From there, we will quantify these air movements by breaking down the specific lung volumes and capacity. You can expect the examiner to test your ability to differentiate these on a graph. So we will make sure you know them inside out. Next, we will cross the boundary into the blood, focusing on gas exchange at the alveolar capillary membrane, where simple diffusion dictates how oxygen enters and carbon dioxide exits our system. Once those gases cross the membrane, they must travel. We will detail the transport of oxygen and carbon dioxide in blood. This leads us directly into a high-yield favorite for examiners: hemoglobin structure, function, and the famous oxygen dissociation curve. You will learn exactly what makes this protein shift its affinity for oxygen depending on the tissue environment. But how does your body know when to change its breathing rate? We will look at the control of breathing through both neural and chemical regulation. This involves a sophisticated feedback loop driven by peripheral and central chemoreceptors, which constantly sample your blood and cerebrospinal fluid for changes in pH and gas concentrations. To see these systems in action, we will examine the physiological response to hypoxia, hypercapnia, and exercise, showing how the body adapts to low oxygen, high carbon dioxide, and intense physical demand. Finally, we will tie all these concepts together with high-yield clinical correlations, exploring the physiological impacts of high altitude asthma and COPD. Get ready, because we are covering it all at examiner depth. All right, let us start at the very beginning, and I mean truly at the beginning, because the lungs do not operate in isolation. They are part of a complete system, and I want you to understand that system structurally before we talk about any of the physiology. The respiratory system has one overarching job: to allow oxygen to move from the air outside your body into the blood, and to allow carbon dioxide to move from the blood back out into the air. Everything, every structure, every cell type, every reflex is in service of that one exchange. Now the system is divided into two functionally distinct zones. The first is the conducting zone. This is everything from your nose down to the terminal bronchioles. You can see in the image, the conducting zone starts from the nose and it ends at the terminal bronchiole. That means from here up until this place. This is the conducting zone. And here's the critical thing you must understand about the conducting zone. No gas exchange happens here. None. The air in the conducting zone is just being transported from outside to the exchange surface. It is being warmed, it is being humidified, it is being filtered, but it is not exchanging gases with blood. The second zone is the respiratory zone, which begins at the respiratory bronchiole, continues through the alveolar duct. As you can see here, this is the alveolar duct, and ends at the alveolar sacs and individual alveoli. This is where the actual business of respiration occurs. Let me walk you through the anatomy sequentially. Air enters through the nasal cavity or the mouth. This is the nasal cavity. In the nasal cavity, it gets warmed by a rich blood supply, humidified by the mucous membrane, and filtered by mucus and cilia. From there, it passes through the pharynx. This is pharynx. The air will travel like this. Then it will go to larynx, where the vocal cords are, and into the trachea. The trachea is a fascinating structure. This part is the trachea that we're talking about. It is held open by a C-shaped cartilaginous ring. These are the rings. 1, 2, if you count them all, there will be roughly 16 to 20 rings of them stacked like a stack of horseshoes. So the trachea looks like this, C-shaped. The open part, this portion, faces posteriorly and the gap is bridged by a muscle called the trachealis. Why is there a gap? Why it's not a complete circle like this? Because the esophagus sits right behind the trachea. Let's say this is trachea, and just behind trachea, let's say this is esophagus. As esophagus sits right behind trachea, the cartilaginous ring remains open. Because when you swallow a large bolus of food, let's say this is a bolus of food and you are swallowing, the esophagus needs to expand. It will expand like this. If the trachea had complete rings all the way around, swallowing would be compromised. At the level of sternal angle, which anatomists label T4, T5, we mean this region, the trachea divides. This division point is called carina. From the carina, you get a right primary bronchus, we mean this one, and a left primary bronchus, this one. And here is something IMAT loves to test. You need to remember the right primary bronchus is shorter, wider, and more vertical than left. Whenever any object is aspirated, let's say this is the object, whenever we take food, the food passes through esophagus. But whenever the food or some object is not following the path of esophagus, rather coming into trachea, we call that aspiration. And whenever aspirated objects come inside trachea, for example this, it preferentially goes to the right primary bronchus due to its shorter, wider, and vertical build compared to the left one. And that is written in the exam traps. Don't confuse the right primary bronchus with the left one. Just changing the word right to left will change the complete perspective. The airways then branch further. This was the primary or main bronchus. To be more precise, right main bronchus. They divide into secondary bronchus. Let's color code it. This portion is the secondary bronchus. This is number 1. Let's say this is number 2 and this is 3. So the right primary bronchus will have 3 lower bronchus or lower bronchi, which will go to 3 lobes. Let's say this is the right lung. Lung is divided into 3 lobes, 1, 2, 3, and each lower bronchus will get inside lobes of the right lung. This secondary lower bronchus will keep dividing to make these bronchi, which are called segmental bronchi, or also called tertiary bronchi. This segmental bronchi will start branching out as bronchioles, such as this, seen in the image. These bronchioles will further keep dividing to branch out as terminal bronchioles, which will eventually become respiratory bronchioles. To wrap it up, we need to rebuild the respiratory tree so that you don't get confused. First, this is the trachea that we have drawn. From trachea, you will have bronchus. For convenience, we are just drawing the right side. So this would be the right main bronchus. From right main bronchus, you will have lower bronchus 1, 2, and 3. So 3 lower bronchi. Bronchi is the plural and bronchus is singular. That's why I'm interchanging the words every now and then. So this is the trachea. We set T for it. This is the main bronchus. Which one is right? Then from main bronchus, we have lower bronchus 1, lower bronchus 2, lower bronchus 3. This lower bronchi will further divide to give us segmental bronchus. When you will go into the undergrad or your med schools, you will know further in details about them, but we are superficially covering it as much as IMAT demands from us. So from left bronchus, it will further divide into segmental bronchus. They have particular numbers, but we are just being approximate here. So let's say this is a segmental bronchus, more like this. And from one segmental bronchus, you will have bronchioles like this. And from bronchioles, you will have terminal bronchioles branching out like this. From terminal bronchioles, you will eventually have respiratory bronchioles here. and each respiratory bronchiole will be connected with alveoli. And together, the alveolus are called alveoli. That means alveolus is singular and alveoli is plural. The terminal bronchiole that we were talking about, these terminal bronchioles don't have any cartilage. They only have smooth muscle. And they are the last branch of the conducting zone. After that, everything is a part of respiratory zone. The total volume of the conducting zone is approximately 150 milliliters. The number matters enormously because it represents the anatomical dead space. We will come back to that when we discuss alveolar ventilation. So we have covered anatomical dead space, which is approximately 150 ml. And another fact is the lungs themselves contain 300 to 500 million alveoli, and their total surface area is approximately 70 to 100 square meters. To put that in perspective, that is roughly the floor area of a tennis court. Let's say this is a tennis court. Let's say the floor area or the surface area is around 70 to 100 square meter. And if you take all the surface area of the lung and measure it, it will be around the size of a tennis court. This enormous surface area is the engineering solution to the problem of moving enough gas quickly enough to support life. And I will repeat the exam pearls because they are very important for exams. First one is terminal bronchioles are the last conducting structures. They have no cartilage, only contain smooth muscle. The respiratory bronchioles are the first respiratory zone structure that we have seen, and they begin to bear occasional alveolar outpouchings. That is written in the next stage. Why the word occasional here? Because the region is a gradual transition zone rather than an abrupt ending. In biology, the respiratory system doesn't instantly switch from a pipe to a gas sac. It blends smoothly through a structural gradient. Now imagine the very end of our respiratory tree that we have already drawn. I mean, this structure. Now this is our sole point of discussion. This is the respiratory bronchiole. Imagine the very end of our respiratory tree. We mean the respiratory zone that we were discussing in the previous image. We said this is the terminal, this is the respiratory bronchiole, and with each respiratory bronchiole, you will have alveolus attached. That's why the terminal units here are alveoli and singular is alveolus. These are microscopic, delicate, blind-ended air sacs. They're primary units where gas exchange takes place. But for the oxygen to move from the air inside the alveolus into the blood circulating in the pulmonary capillaries, it has to cross a physical barrier. This barrier is the respiratory membrane, an incredibly thin structure measuring only about 0.5 micrometers across, designed specifically to optimize gas diffusion. So as we were saying, alveolus has capillaries attached to it, almost like capillary network that revolves around the alveolus. And between the alveolar sac and the capillaries, you will have a membrane that is called a respiratory membrane. In this image, this part represents the respiratory membrane, and we mentioned it is about 0.5 micrometers across. If we look closely at the architecture of the alveolar wall, it is lined by two distinct types of epithelial cells called pneumocytes. The first type, type 1 pneumocytes, are extremely flat, squamous cells. Because they are so thin, they cover roughly 95% of the total alveolar surface area, acting as the primary diffusion cells through which gases easily pass. The remaining surface is occupied by type 2 pneumocytes. These are cuboidal. Let's say this is a type 2 pneumocyte, and they have two massive responsibilities. First, they act as the progenitor cells, meaning when type 1 cells are injured, type 2 cells proliferate and differentiate to make these type 1 cells. Second, they synthesize and secrete a crucial substance called surfactant. Now, why do we need surfactant? Let's look at the physics of the alveolus. The inner surface of the alveolus is lined with a thin layer of water. Imagine like this. This is the thin layer of water inside alveolus. Water molecules are highly attracted to each other, creating an inward pulling force known as surface tension. According to the law of Laplace, the collapsing pressure generated inside a spherical structure, we denote that as this P, directly proportional to twice the surface tension and inversely proportional to the radius r, written mathematically as P is equal to 2T over r. You need to understand that all alveoli are not same in sizes. There can be larger ones, there can be smaller alveoli as well. So now if the surface tension were constant everywhere, a smaller alveolus with a tiny radius r would develop a massively high internal pressure compared to a larger alveolus, because r is inversely proportional to the pressure. The less the radius, the more the pressure. That's why pressure will be very high in case of smaller alveolus, and larger alveoli will have small pressure. So let's draw one large alveolus, one small alveolus. The pressure inside the large alveolus is very small. The pressure inside the small alveolus is very high. We just have calculated it with the law of Laplace. This pressure gradient will cause the air to flow from high pressure, that means smaller alveolus, to low pressure, that means large alveolus, like this, causing small alveoli to empty completely into larger ones. This would lead a widespread alveolar collapse. The smaller one will eventually collapse like this, and this pathological state is called atelectasis. To prevent this, the type 2 cells, the type 2 pneumocytes we were discussing, secrete surfactant, which is a specialized phospholipid, specifically DPPC, which stands for dimethyl methyl phosphatidylcholine. You don't need to remember that mouthful word. Just know that surfactant is mainly a specialized phospholipid. The surfactant works by inserting itself between the water molecules. So this was the thin water layer revolving around the alveolus. When surfactant, let's say surfactant comes here, it inserts between the water molecules and it breaks the cohesive bonds and reduces the surface tension. More importantly, surfactant molecules become much more concentrated in smaller alveoli. That means the smaller one will have more surfactants compared to the larger ones. This means surfactant reduces surface tension dynamically, equalizing the pressure across both small and large alveoli, stabilizing them, and preventing smaller ones from collapsing into larger ones. By reducing the surface tension component of elastic recoil, surfactant drastically increases lung compliance, making the lungs significantly easier to expand. Now let's tie this entire concept directly back to the key terms and exam traps you will face on the test. When you review your notes, remember the strict definition of the alveolus. It is the terminal air sac and the primary gas exchange unit of the respiratory system. The barrier separating air and blood is respiratory membrane, which is roughly 0.5 micrometers thick. For the exam, you must clearly distinguish between the two key cellular features of the alveolus. Type 1 pneumocytes are flat, squamous cells covering 95% of the alveolar surface, optimized as primary diffusion cells. In contrast, type 2 pneumocytes are cuboidal cells that secrete surfactant and serve as the progenitor for type 1 cells. A classic example is to memorize that type 2 cells also regenerate type 1 cells after injury. And surfactant is mainly a specialized phospholipid, mainly DPPC. It reduces surface tension and prevents alveolar collapse. Let's look at the mechanism and sequence of the surfactant action through the lens of physics. The law of Laplace states that pressure equals 2T divided by R. Under normal conditions, surfactant equalizes pressure between small and large alveoli. If no surfactant is present, the sequence is clear. Small alveoli empty into large ones due to Laplace's law, leading directly to atelectasis. When surfactant is present, surface tension is reduced, creating a stable, equal pressure across all alveoli. By minimizing the surface tension component of elastic recoil, surfactant effectively increases lung compliance. Finally, look out for this specific exam traps designed to catch you off guard. First, do not confuse shapes and functions of the pneumocyte. Type 1 cells are thin and responsible for gas exchange, while type 2 cells are cuboidal and responsible for surfactant production. Second, understanding the clinical correlation with neonatal respiratory distress syndrome or neonatal RDS. This condition is caused by surfactant deficiency that typically occurs before 34 to 36 weeks of gestation. Gestation means the progression of pregnancy, as the fetal lungs have not yet produced enough DPCC to sustain independent breathing. Third, never forget the biochemical nature of this molecule. Surfactant is a phospholipid, specifically DPPC, and it's absolutely not a protein. Examiners love to test whether you know it's a lipid-driven compound rather than a peptide. To understand how we breathe, we have to look at a fundamental rule of physics, and that is fluid always move from an area of high pressure to an area of low pressure. Breathing is essentially the creation of a pressure gradient between the atmosphere and the microscopic alveoli inside our lungs. But how do we change pressure inside a closed biological container? We do it by changing its volume utilizing Boyle's law. Let's look at the active phase of this process, inspiration. When you decide to take a breath in, your brain sends a signal to your primary muscle of respiration, the diaphragm. At rest, when you decide to take a breath in, your brain sends a signal to your primary muscle of respiration, the diaphragm. At rest, the diaphragm is dome-shaped, curving outward into the thoracic cavity. When it contracts, it flattens out and moves down. That's why this down arrow is given. This downward movement directly increases the thoracic volume in the vertical dimension. That means the volume will increase like this. Simultaneously, your brain activates the external intercostal muscle, the muscles that are found in between the ribs here. When these fibers contract, they elevate the ribs, pointed by these up arrows here. Think of this like lifting a bucket handle. Let's say this is a bucket, we have handle. If you lift the handle will come over here and it will increase the volume. And put that into perspective, in case of ribs, the ribs swing upward and outward. That means which drastically increases both the anterior, posterior, and the lateral dimension of the chest cavity. Because the lungs are structurally bound to the chest wall, when the thoracic volume increases, the lung volume must increase along with it. As the lungs expand, the pressure inside the alveoli falls below atmospheric pressure by about 3 mm of mercury. Because alveolar pressure is now lower than the air outside, a pressure gradient is established, and air naturally flows down its gradient directly into the lungs. And what happens when you breathe out during normal, quiet breathing? This process is completely passive. Quiet expiration does not require any active muscle contraction. Instead, the diaphragm and the external intercostal simply relax. The diaphragm moves back up to its dome shape. You see, the shape has become like a dome right now, and the ribs drop back down. The lungs and the chest wall possesses a natural spring-like elastic recoil. As they snap back to their resting state, the overall thoracic and lung volume decreases. According to Boyle's law, a decrease in volume causes the internal pressure to rise. That means volume is going down, pressure is increasing inside, and alveolar pressure increases above atmospheric pressure, reversing the gradient and forcing air out of the lungs. In this case, the air will come out of the lungs and go into atmosphere. However, if you need to blow out candles or clear your airways via forced expiration, this becomes an active process. To forcefully decrease thoracic volume even further, your body recruits the internal intercostal and abdominal muscles. Internal intercostals are the muscle groups that are just under the external intercostal muscle layer. When these internal intercostal muscles and the abdominal muscles are recruited by our body, they get contracted, which violently pull the ribs down and push the diaphragm upward. And throughout this entire cycle, the lungs are kept from collapsing due to unique pressure dynamic within the pleural space. The intrapleural pressure, that is the pressure inside the fluid-filled space between the lungs and the chest wall. I mean this region, this intrapleural pressure is always subatmospheric, sitting at roughly -5 centimeter water pressure at rest. This negative pressure acts like a vacuum, counteracting the natural inward elastic recoil of the lungs and keeping them pinned against the chest wall, fully expanded. If this sealed system is breached, such as by a puncture wound to the chest wall, air enters to the pleural space, a condition known as pneumothorax. The moment air pushes in, the intrapleural pressure equalizes with atmospheric pressure. The lung instantly loses its transmural pressure gradient, and its unchecked elastic recoil causes the lung to completely collapse. Now let's map this exact physiological concepts directly to the text and terminology on your slides so you are fully prepared for the exam. Starting with the core definition, breathing is defined as the creation of a pressure gradient between the atmosphere and the alveoli, specifically driven by volume changes. When analyzing the mechanism and sequence, the exam will expect you to categorize the phases precisely. Inspiration is an active process. The sequence is diaphragm contracts and moves down, increasing thoracic volume vertically. External intercostals contract, elevating the ribs via bucket handle movement to increase the anterior, posterior, and lateral dimensions. The resulting increase in lung volume causes alveolar pressure to fall approximately 3 mm mercury below atmospheric pressure, causing air to flow in. Quiet expiration is a passive process where the diaphragm and external intercostal relax. The elastic recoil of the lung and the chest wall causes volume to decrease, alveolar pressure to rise, and air to move out. Forced expiration is an active process where the internal intercostals and abdominal muscles contract. Just for clarity, I want to briefly describe what elastic recoil is. Elastic recoil of the lungs is the intrinsic physical tendency of the lungs to spontaneously deflate, shrink inward, and return to their resting volume after being stretched by inhalation. This elastic recoiling tendency is caused by alveolar surface tension, and as the fibers are elastic, it is also responsible for that elastic recoil. Pay extremely close attention to these specific exam traps. A favorite trap is confusing the active and passive states. Remember that quiet expiration is purely passive, driven by elastic recoil only, with absolutely no muscle contraction involved. Do not swap your intercostals. Internal intercostals are utilized for forced expiration, while external intercostals are utilized for inspiration. Additionally, note that accessory muscles, specifically the sternocleidomastoid and the scalenes, are exclusively recruited during deep or forced inspiration, not during quiet breathing. Finally, memorize these high-yield exam pearls regarding pleural dynamics. Intrapleural pressure is always subatmospheric, measuring around -5 cm of water at rest, which is the mechanical force that keeps the lungs expanded. If a patient develops a pneumothorax, air enters pleural space, causing the lung to lose its transmural pressure gradient, which leads directly to a collapsed lung. To understand how our lungs function dynamically, we need a way to measure the air moving in and out of them. This is where spirometry comes into play. Imagine a patient breathing into a machine that traces their breaths as a continuous wave on a graph. Like seen in the image, the person is breathing, and on the monitor, we see the graph being generated in real time. When you are sitting quietly, breathing normally without thinking about it, each breath moves a relatively small volume of air. This baseline wave represents your tidal volume, or TV here, which typically averages about 500 ml of air. but our lungs have massive reserves beyond a quiet resting breath. Imagine you are at the end of a normal quiet inhalation, and I tell you to breathe in as deeply as you possibly can. That extra volume of air you can force into your lungs above your normal tidal breath is your inspiratory reserve volume, which sits at a substantial 3000 milliliter. If we want to see it on the graph, then it will start from here and to this, which is inspiratory reserve volume. On the flip side, let's say you breathe out normally, hitting the bottom of your quiet tidal wave, and then I ask you to actively force out every single drop of air left in your lungs. That extra air you just pushed out below your normal tidal breath is your expiratory reserve volume, or ERV, averaging around 1200 milliliter. Now here is a critical concept. No matter how hard you try, you can never completely empty your lungs. Even after the most forceful maximal expiration, a specific amount of air always remains trapped inside your airways and alveoli to prevent them from completely collapsing. This non-movable air is your residual volume, or RV, which is roughly 1200 ml. Because spirometry relies entirely on the actual movement of air into and out of a machine, this trapped residual volume cannot be measured by a standard spirometric test. When we start combining these individual foundational volumes together, we get what are known as lung capacities. For example, if you add together your resting tidal breath, this portion, and that massive extra inhale you take, this portion, you get your inspiratory capacity, or IC. Mathematically, IC is equal to PV plus IRV. This gives you a total of about 3500 ml. Next, look at the air left in your lungs at the exact moment you finish a normal quiet exhalation. This is a crucial baseline known as functional residual capacity, which equals your expiratory reserve volume plus that trapped residual volume. This together averages around 2400 ml and represents your resting and expiratory volume. If you want to know the absolute maximum amount of movable air your lungs can possibly handle, we look at your vital capacity, or VC. This is the entire volume of air you can forcefully exhale after taking the deepest possible inhalation. It combines your entire reserve and resting volumes. So VC would be equal to tidal volume, EV, plus inspiratory reserve volume plus expiratory reserve volume, and this comes out to roughly 4700 ml. Finally, if you want to know the absolute total volume of air your respiratory system can hold, including the air you can move and the air that is permanently trapped, you look at your total lung capacity, or TLC. You can calculate this by taking your maximum movable air and adding the trapped baseline. That would be vital capacity plus residual volume, RV. This leaves you with a grand total of approximately 5900 ml of air. Now let's anchor this exact physiological parameters, mathematical relationships, and values to the specific terms on your slides so you can effortlessly spot the correct answers on your exam. Starting with the core definition, spirometry is a tool that strictly measures air movement. Because of this mechanical limitation, residual volume, or RV, cannot be measured by standard spirometry. Let's break down the key features table, memorizing the normal values and components for each volume and capacity. Tidal volume has a normal value of 500 ml and represents one normal breath. Inspiratory reserve volume has a normal value of 3000 ml and is defined as the extra air inspired above tidal volume. Expiratory reserve volume has a normal value of 1200 milliliter and is the extra air expired below tidal volume. Residual volume, RV, has a normal value of 1200 ml and presents the air that remains in the lungs after maximum expiration. Remember, this is not measurable by spirometry. Vital capacity sits at 4700 ml and is the sum of PV plus IRV plus ERV. Functional residual capacity is 2400 ml and is calculated as ERV plus RV, representing the resting and expiratory volume. Inspiratory capacity is 3500 ml and is composed of TB plus IRB. Total lung capacity stands at 5900 ml and is the absolute total calculated as VC plus RV. When analyzing the spirometric graph, pay close attention to how these selections align. Notice how the small resting waves of tidal volume, this region, sit directly between the massive upward peak of vital capacity and the downward dip of expiratory reserve volume, while residual volume sits as a completely isolated block at the very bottom of the lung architecture here. Finally, guard yourself against these classic exam traps. The highest yield trap on this topic concerns measurability. Any value that includes residual volume, which means RV itself, functional residual capacity, and total lung capacity, cannot be measured by spirometry alone. Conversely, vital capacity does not include residual volume, which means it is completely measurable by spirometry. Do not mix up your capacities. FRC represents your resting and expiratory lung volume, which is mathematically and conceptually distinct from your inspiratory capacity, IC. Ensure you do not swap those definitions or their corresponding components when faced with a quick calculation question. When we evaluate a patient with lung disease using spirometry, we look at how much air they can forcefully exhale and how fast they can do it. There are two primary clinical metrics we must establish first. The first one is forced expiratory volume in 1 second, FEV1, which measures the volume of air cleared during the very first second of a forced breath. And the second one is forced vital capacity, which is the total volume of air exhaled across the entire breath. By looking at the ratio of these two values, written as FEV1 divided by FVC, we can definitely differentiate between two massive categories of pulmonary pathology: obstructive lung disease and restrictive lung disease. Let's look from first principles at what happens in an obstructive pattern, such as asthma or chronic obstructive pulmonary disease, COPD. The fundamental problem here is an increase in airway resistance, meaning the airways are narrow. It's a normal width airway, but in this obstructive disease, the airway becomes narrower, or it can be floppy or physically blocked as well. When a patient tries to exhale forcefully, the air encounters tremendous resistance, making it incredibly difficult to get the air out quickly. In normal case, the air will get out in normal pace. But when a patient has obstructive condition, he will struggle a lot trying to push out air from this narrow tube. Because of this expiratory flow limitation, the volume of air they can clear in that crucial first second, that is FEV1, plummets drastically. The total capacity to eventually push air out, the FBC we mean, this can also decrease, but it doesn't drop nearly as much as the FEV1. Because the formula was FEV1 divided by FVC, and the numerator falls drastically while the denominator is relatively preserved, this ratio drops significantly below normal, often falling under 0.7. Over time, because the air cannot clear easily during expiration, it gets left behind inside the alveoli. This leads to air trapping. Because air is trapped at the end of every breath, the baseline volumes expand, meaning the total lung capacity, or TLC, will be completely normal or even significantly increased. Now let's compare that with restrictive pattern, which we see in conditions like pulmonary fibrosis or thoracic deformities like kyphoscoliosis. The primary issue here is not the airways, it is the lung parenchyma or the chest wall itself. Parenchyma means the cells of the lungs, for simplification's sake. In this restrictive condition, the lungs become stiff, hard, or restricted from expanding properly. Because the lung cannot physically expand to take in air, the total lung capacity is always reduced. When it comes to exhalation, the airways themselves are perfectly clear, so the patient has no trouble pushing air out fast. However, because they couldn't fill their lungs in the first place, they have much less total air to move. As a result, both FEV1 and FVC decrease together in a balanced proportional manner. Because the numerator and the denominator drop symmetrically, the mathematical ratio of FEV1 and FVC remains completely normal, or it can even increase above 0.7. To refine our clinical picture, we also test for reversibility by administering a bronchodilator. Bronchodilators are the medications which will dilate the airway. Let's say the airway was narrowed. When we apply bronchodilator here, it will be dilated. One common example is albuterol, and we do this to see if the airway obstruction resolves. In a patient who has asthma, the airway narrowing is primarily driven by smooth muscle bronchoconstriction and inflammation, which means the obstruction demonstrates partial or complete reversibility on spirometry. In a patient with COPD, particularly emphysema, the structural architecture of the lung is permanently destroyed. The loss of alveolar walls means the lungs lose their natural elastic recoil, turning into a highly compliant floppy lung that easily traps air. Because this is a permanent structural destruction, a bronchodilator shows minimal to no reversibility, highlighting the fixed nature of the obstruction. Now let's tie these distinct patterns directly back to the exact terminology, values, and diagnostic criteria outlined on your slide, so you can easily navigate any case study on the exam. Starting with the core definitions, FEV1 is defined as the forced expiratory volume in 1 second, and FVC is the forced vital capacity. The diagnostic threshold for the FEV1 and FVC is absolute. A ratio less than 0.7 defines an obstructive pattern, while a normal ratio or a ratio greater than 0.7 indicates restrictive pattern. Let's review the key features table to lock in the contrasting profile for each pattern. For the obstructive pattern, the ratio of FEV1 and FVC will be less than 0.7. The TLC will be normal or high due to air trapping. Reversibility is partial in asthma or absent in COPD. Classic clinical examples include asthma and COPD. For the restrictive pattern, the ratio stays normal or it can even be greater than 0.7. The TLC is characteristically reduced. Reversibility is minimal. Classic clinical examples include fibrosis and hyposcoliosis. Make sure you memorize these high-yield exam traps to avoid dropping easy points. In a restrictive disease, do not assume a drop in this ratio means an obstructive ratio. The ratio can actually be elevated, often exceeding 0.8, because both individual values fall proportionally. Remember the classic clinical distinction. Asthma represents a reversible obstruction, whereas COPD represents an irreversible obstruction. Additionally, in emphysema, the TLC may be increased specifically due to air trapping. This occurs because the lung experiences increased compliance and a loss of elastic recoil, creating a highly distensible but inefficient respiratory unit. Finally, let's consolidate the core mechanical principles from our exam pearls section. In fibrosis, compliance decreases, creating a stiff lung that restricts expansion, which leads directly to reduced TLC. In emphysema, compliance increases, creating a floppy lung that leads to severe air trapping, which clinically manifests as an increased TLC. Don't get confused with the word compliance. It simply means the ability of the lungs to stretch and expand. To understand how gases move between the air we breathe and the blood in our bodies, we must look at the rule of gas physics. The first foundational principle is Dalton's law, which states that the total pressure of a mixture of gases is simply the sum of individual pressures exerted by each component gas on its own. We call these individual pressures partial pressure. The second principle is that gases are simple passive travelers. They always diffuse from area of high partial pressure to an area of low partial pressure. No active transport, no energy required. It is pure down the gradient diffusion. Now let's look at the actual numbers as air travels from the outside world into our bodies. Out in the dry atmosphere, the partial pressure of oxygen, or PO2, is high, about 160 millimeter of mercury, while the partial pressure of carbon dioxide is practically negligible at 0.3 millimeter of mercury. But the moment that air enters your respiratory tract, everything changes. Your upper airways are warm and moist, which means water vapor is immediately added to the incoming air. This means when the air gets inside through these pipes, it has vapor in it, and the vapor, which is basically water, puts its own pressure, which is around 47 millimeter of mercury. And this actively dilutes the incoming gases. Because of this dilution and the fact that fresh air constantly mixes with old air left in the lungs, the oxygen pressure drops. By the time the air reaches the alveoli, the alveolar PO2 is stabilized at 100 millimeter of mercury, and the alveolar partial pressure of carbon dioxide sits at 40 millimeter of mercury. That's written here. Now look at the blood arriving at the lungs. This is the capillary that we have drawn, and blood is arriving from left to right in this particular image. Deoxygenated venous blood is pumped from the right side of the heart. This is the heart. Heart has 4 chambers. This is right atrium, right ventricle, left atrium, left ventricle. From right ventricle, deoxygenated blood is pumped. This is pumped from the right ventricle, and it comes to pulmonary artery. From pulmonary artery, it directly flows into pulmonary capillaries, wrapping around the alveoli. This blood has come just from the tissues, so it is starving for oxygen and loaded with waste. Its partial pressure of oxygen is quite low, at 40 millimeter of mercury, while its partial pressure of carbon dioxide is elevated at 46 millimeter of mercury. Look at the beautiful pressure gradients that are instantly created across the tiny 0.5 micrometer respiratory membrane. If you imagine we have an alveolus here, so this place is the respiratory membrane, and in this respiratory membrane we already have a pressure gradient right now. For oxygen, you have a gradient of 100 millimeter mercury in the alveolus versus 40 millimeter mercury in the capillary. Oxygen naturally and rapidly diffuses out of the alveolus into the blood, shown in this specific segment. For carbon dioxide, you have 46 millimeter mercury in the blood versus 40 millimeter of mercury in the alveolus. It travels down its gradient, diffusing out of the blood and into the alveolus to be exhaled. By the time the blood leaves the pulmonary capillaries and enters the pulmonary vein as oxygenated, we can imagine this portion as the pulmonary vein. It has achieved perfect equilibrium with the alveolar air, which was 100 millimeter mercury for oxygen and 40 millimeter mercury for carbon dioxide. And it leaves the lung with a partial pressure of oxygen having 100 millimeter and partial pressure of carbon dioxide having 40 millimeter. But why does carbon dioxide equilibrate so perfectly when its pressure gradient is only 6 millimeter mercury? Here 46 and here 40. That means only 6 is the pressure gradient. But you see the pressure gradient of oxygen is around 60 millimeter mercury. So there's a huge gradient difference, but still carbon dioxide equilibrate perfectly. To answer this question, you need to get introduced with Fick's law of diffusion. The equation is where V gas stands for the rate of gas transfer, area means the surface area of the membrane, a diffusion coefficient, partial pressure gradient, and membrane thickness. So we can say rate of gas transfer V gas is proportional to the surface area of the membrane, diffusion coefficient, and partial pressure, but inversely proportional to membrane thickness. Crucially, the diffusion coefficient is directly dependent on the solubility of gas. Carbon dioxide is an absolute superstar when it comes to solubility. It is 20 times more soluble in water and liquid membranes than oxygen is. Because it dissolves so easily, carbon dioxide diffuses 20 times faster than oxygen, meaning it requires a much smaller pressure gradient to move just as effectively. This has massive clinical implication. If a patient develops a pathology that thickens the respiratory membrane, like pulmonary fibrosis, oxygen is always the very first gas to suffer. Because oxygen lacks the solubility advantage, its diffusion is impaired early, causing blood oxygen levels to drop. Carbon dioxide diffusion, thanks to its 20-fold solubility advantage, remains unbothered for a long time, only becoming impaired much later when the disease becomes exceptionally severe. Now let's connect this entire physiological narrative directly to the key facts, figures, and definitions listed on your slide so you can spot them instantly on test day. Starting with our core definitions. Dalton's law states that total pressure equals sum of partial pressures of component gases. Gas diffusion strictly occurs from high partial pressure to low partial pressure. Let's carefully memorize the exact partial pressure values across different locations from the key features table. In the dry atmosphere, partial pressure of oxygen is 159 mm mercury and partial pressure of carbon dioxide is 0.3 mm of mercury. In alveolar air, partial pressure of oxygen drops to 100 mm mercury and partial pressure of carbon dioxide is 40 mm mercury. In pulmonary artery carrying venous blood, partial pressure of oxygen is 40 mm of mercury and partial pressure of carbon dioxide is 46 mm of mercury. In the pulmonary vein carrying arterial blood, partial pressure of oxygen equilibrates back to 100 mm and partial pressure of carbon dioxide returns to 40 mm of mercury. At the level of tissue cells, that means when the blood reaches the tissue, partial pressure of oxygen is less than 5 mm of mercury as cells consume it, and partial pressure of carbon dioxide is roughly 46 mm of mercury. When studying the mechanism and sequence, map out the directional flow. Oxygen moves from alveolar partial pressure of oxygen 100 to a blood which has partial pressure of oxygen of 40, diffusing into the blood. Carbon dioxide moves from a blood which has carbon dioxide partial pressure of 46 to an alveolar partial pressure of carbon dioxide 40 region, diffusing out to the alveolus. Pay critical attention to these high-yield exam traps. Do not fall for the trap of setting alveolar partial pressure of oxygen to atmospheric levels. Alveolar partial pressure of oxygen is 100 mm of mercury, not 159. Because water vapor at 37 degrees centigrade exerts a pressure of 46 mm of mercury, which directly dilutes the alveolar gas. Understand the true driver of diffusion speed. Carbon dioxide diffuses 20 times faster than oxygen due to its massive solubility advantage. Absolutely not because of its molecular weight. In patients with thick membrane disease, remember the presentation timeline. Oxygen exchange is impaired first, while carbon dioxide exchange is impaired late. Finally, lock in the mathematical relationships from our exam pearls. Fick's law states that V gas is proportional to area times diffusion coefficient partial pressure difference, all divided by thickness. Diffusion coefficient is directly proportional to solubility. Carbon dioxide is 20 times more soluble than oxygen in water, easily bypasses modest drops in its pressure gradient. Okay, remember the oxygen scenario where oxygen diffuses across the respiratory membrane into the pulmonary capillaries. This is alveolus and this is the capillary. Oxygen comes from here to the capillaries. When it comes into the pulmonary capillaries, it faces a major hurdle. It needs to travel through the bloodstream to reach the tissues. Oxygen is notoriously poorly soluble in water, and if we relied entirely on the plasma to carry dissolved oxygen, we could only transport a meager 1.5% of what our body actually needs. This baseline limitation is governed by Henry's law, which states that the amount of dissolved gas in a liquid is directly proportional to its partial pressure. Our body utilizes a specialized packaging system inside our red blood cells, hemoglobin. By chemically packing oxygen away, hemoglobin successfully transports the remaining 98.5% of the total oxygen in our blood. Let's dissect the molecular architecture of this biochemical vehicle from first principles. Hemoglobin is a complex tetrameric protein, meaning it is built from 4 distinct polypeptide globin chains. 1, 2, 3, and 4. These are polypeptide globin chains. If we look at normal adult hemoglobin, known as hemoglobin A, it consists of a pair of alpha chains and a pair of beta chains. Tucked safely inside the folds of each of these 4 globin chains is a non-protein prosthetic molecule called a heme group. These are the heme groups shown in the image. At the absolute center of every single heme group sits an atom of iron. The brown-colored particles or dots are symbolized as iron here. But the state of that iron matters tremendously. For oxygen to bind, the iron must be maintained strictly in its reduced ferrous state, written as Fe2+. Think of it this way. Fe2+ is the only functional state that allows for binding. If the iron gets oxidized into the ferric state, written as Fe3+, it transforms into a dysfunctional compound called methemoglobin. Methemoglobin is completely incapable of binding oxygen, rendering that specific site useless. Because there are 4 heme groups per hemoglobin molecule, 1, 2, 3, 4. Each individual hemoglobin vehicle can carry a maximum load of 4 oxygens at any given time. Now let's look at how this architecture changes dynamically before birth. A developing fetus cannot breathe air directly. It has to physically strip oxygen away from the mother's blood across the placenta. Let's say this is mother. Inside mother's womb, we have fetus over here. It is connected with mother through placenta. To achieve that, the fetus utilizes a distinct structural variant called fetal hemoglobin or hemoglobin F. Instead of the standard adult configuration, hemoglobin F is built from 2 alpha chains and 2 gamma chains. This subtle change completely alters its behavior. The gamma chains have a significantly lower affinity for a regulatory molecule in our blood called 2,3-BPG. Because 2,3-BPG normally binds to adult hemoglobin to help it release oxygen, a molecule that binds 2,3-BPG less tightly will hold onto oxygen much more fiercely. This structural adaptation shifts the entire oxygen hemoglobin dissociation curve to the left. We will know more about the oxygen hemoglobin dissociation curve in detail later. That means when hemoglobin F has higher affinity for oxygen, this creates a physiological gradient across the placenta. Let's say this is placenta and this is the fetus. As maternal hemoglobin A moves into an environment where it wants to give up oxygen, the neighboring fetal hemoglobin F acts like a molecular vacuum, extracting that oxygen cleanly to fuel the growing fetus. But this highly sensitive binding site is also vulnerable to a lethal molecular impostor, carbon monoxide, written as CO. Carbon monoxide is terrifying because it targets the exact same binding site on the heme iron as oxygen does, but it binds with an astounding 25 times greater affinity. The moment carbon monoxide occupies a heme site, let's say carbon monoxide sits in here, it forms carboxyhemoglobin, which is completely non-functional for oxygen transport. To make matters worse, it creates a double toxicity effect. When carbon monoxide locks onto 1 or 2 of the 4 heme sites, it alters the conformation of the remaining vacant sites on that same hemoglobin molecule, forcing them into a high affinity state. This causes a massive left shift in the oxygen dissociation curve for the remaining hemoglobin sites. Consequently, the hemoglobin becomes incredibly stingy. Not only it can carry less oxygen because carbon monoxide is blocking the seats indicated here, but it also refuses to release the tiny bit of oxygen it does manage to carry when it reaches the starving tissue. Now let's log the structural variations, biochemical definitions, and competitive mechanisms directly to the terms on your slides to keep your exam performance flawless. Starting with our core definition. Hemoglobin is a tetrameric protein containing 4 heme groups where each individual heme binds to exactly 1 molecule of oxygen. For functional gas transport, oxygen binds only when iron is in its ferrous state. If it is oxidized to the ferric Fe3 state, it becomes methemoglobin, which is strictly non-functional. Let's review the key features and transport metrics from the slide text. Adult hemoglobin is composed structurally of 2 alpha and 2 beta chains. Fetal hemoglobin is composed structurally of 2 alpha and 2 gamma chains. Because the gamma chains bind 2,3 BPG less, hemoglobin F exhibits left-shifted oxygen dissociation curve. More on it later. And this higher oxygen affinity allows hemoglobin F to effectively extract oxygen from maternal hemoglobin A across placenta. Oxygen transport values are highly specific. Roughly 98.5% travels bound to hemoglobin, while only 1.5% travels dissolved in plasma, a physical limit dictated by Henry's law. Pay critical attention to these high-yield exam traps. Do not forget the oxidation states. Fe2 is functional, whereas Fe3 is methemoglobin and cannot carry oxygen. When analyzing carbon monoxide poisoning, remember that carbon dioxide binds hemoglobin at the exact O2 site approximately 25 times greater affinity. When analyzing carbon monoxide poisoning, remember that carbon monoxide binds hemoglobin at the exact O2 site with approximately 250 times greater affinity, generating non-functional carboxyhemoglobin. For the exam, memorize the double toxicity mechanism of carbon monoxide. It blocks binding seats and simultaneously causes a severe left shift of the oxygen dissociation curve for any remaining hemoglobin sites, completely halting tissue delivery, which is known as Haldane effect. Finally, cement these foundational concepts from our exam pearls. The left-shifted oxygen dissociation curve of hemoglobin F represents a higher affinity profile, which is the exact mechanical reason why fetal extraction of oxygen from maternal hemoglobin across the placenta is possible. Always separate capacity from saturation. The overall hemoglobin concentration determines the absolute oxygen carrying capacity of blood, whereas the ambient partial pressure of oxygen, or PO2, is the sole factor that determines hemoglobin saturation. Now we reach perhaps the single most tested concept in respiratory physiology on the oxygen hemoglobin dissociation curve. The curve is not something to memorize, it is something to understand deeply, because every question about it is ultimately asking whether you understand the underlying mechanism. If you plot the partial pressure of oxygen on the x-axis against the percentage saturation of hemoglobin on the y-axis, you don't get a straight linear line. Instead, you get a distinctive sigmoidal S-shaped curve. Why does it take this specific shape? It comes down to a structural property called cooperative bonding. Hemoglobin exists in two basic conformational states. The T state, which is tense, and this tense or T state has low affinity for oxygen. And another state is R state or relaxed state, which has a high affinity. When hemoglobin is completely deoxygenated, that means hemoglobin doesn't have any oxygen. It is locked in the tense state. It takes a relatively high partial pressure of oxygen just to force that very first oxygen molecule to bind. But the moment that first oxygen snaps onto a heme group, it triggers a structural domino effect. The entire tetramer undergoes a conformational shift, breaking salt bridges and relaxing into R state. This relaxation makes it exponentially easier for the second, third, and fourth oxygen molecules to bind. To track where a patient's curve sits, we use a standard physiological benchmark called the P50. P50 is strictly defined as the partial pressure of oxygen at which hemoglobin reaches exactly 50% saturation. Normally, this sits at about 26 to 27 millimeter of mercury. If the curve shifts to the right, you see this one has shifted to the right. It means the P50 value increases. Higher P50 means you need more pressure just to get to half saturation, which reflects a reduced oxygen affinity. This reduced affinity allows hemoglobin to unload oxygen much more easily to the tissues. Conversely, a left shift, indicated here with green curve, means the P50 decreases, reflecting an increased oxygen affinity where hemoglobin holds onto oxygen tightly and unloads less. Let's look at the physics of how the curve's unique shape serves our survival. The curve is divided into two distinct zones, a steep portion and a plateau. This is the steep portion. This steep portion sits at lower pressures, roughly between 20 and 60 millimeter of mercury. This is the exact environment of our systemic tissue. Because it is so steep, a tiny drop in tissue PO2 causes a massive drop in hemoglobin saturation, ensuring that large amounts of oxygen are instantly released right where the cells are actively consuming it. The plateau sits at higher pressures, running from 60 millimeter of mercury all the way up to 100 millimeter, which is the environment of our lungs. Because the curve flattens out here, loading at the lung is safely maintained. Even if a patient experiences moderate hypoxia, that means less oxygen, and their alveolar PO2 drops from 100 down to 70, hemoglobin remains highly saturated at over 90%, acting as a protective safety net. Finally, how does our body shift this curve during real-time physiological stress? Imagine you are exercising hard. Your skeletal muscles are burning glucose, pumping out carbon dioxide, generating heat and releasing lactic acid, which lowers the local pH. Every single one of these factors, increased carbon dioxide, increased hydrogen ions or protons, increased temperature, and increase in the metabolic byproduct 2,3-BPG, acts directly on the hemoglobin molecule to stabilize its tense state. And this triggers a right shift seen in this graph. This classic phenomenon is called Bohr effect. An environment with high carbon dioxide and low pH actively forces hemoglobin to unload more oxygen directly to those active working tissues. On the flip side, if you look at the lungs, opposite happens. Oxygen binds hemoglobin and unloads carbon dioxide, a phenomenon known as the Haldane effect. Now let's anchor this precise curve shifts, behavioral zones and physiological effects directly to the text and diagnostic terms on your slides. Starting with our core definitions. The ODC, oxygen dissociation curve, is a sigmoidal curve relating partial pressure of oxygen to hemoglobin saturation. Its sigmoidal shape is driven entirely by cooperative bonding, which is a structural conformational shift from the tense state to the relaxed state. The benchmark metric P50 is defined as the partial pressure of oxygen at 50% saturation, normally measuring at around 26 to 27 millimeter of mercury, where a right shift strictly increases P50. Let's review the shift dynamics and tissue delivery effects directly from the key features table. An increase in carbon dioxide shifts the curve to the right, releasing more oxygen active tissues. An increase in proton, that means low pH, shifts the curve to the right. This is the Bohr effect, where acidosis unloads oxygen. An increase in temperature shifts the curve to the right, ensuring active or febrile tissues receive more oxygen. An increase in 2,3-BPG shifts the curve to the right, serving as a critical adaptation during chronic altitude or anemia. Carbon monoxide poisoning causes a left shift, combined with a reduced carrying capacity, causing severe oxygen delivery failure. Fetal hemoglobin F versus hemoglobin A represents a characteristically left-shifted curve, optimizing fetal extraction at the placenta. When navigating high-yield exam traps, use these exact rules to keep your points safe. Remember the right shifting factors. Instantly use the classic memory hook, caret, pace, right. This stands for C, carbon dioxide, A, acid, D, BPG, T, temperature, E for exercise. Guard yourself against a major conceptual trap. A right shift means a lower oxygen affinity, which results in better, highly optimized tissue delivery. Absolutely not worse. Note that on the plateau curve, above PO2 of 60 millimeter mercury S8O2. That means saturation of oxygen is remarkably well maintained even when a patient faces moderate hypoxia. Finally, lock in the core structural zones from your exam pearls. The steep portion, this portion, is precisely where tissue delivery occurs because a small drop in PO2 yields a large release of oxygen. The plateau region, this region, ensures that oxygen loading at the lung remains fully stable and secure, even if the ambient alveolar PO2 drops moderately. Our next topic is carbon dioxide transport, bicarbonate, chloride shift, and Haldane effect. Let's build the physiology of carbon dioxide transport completely from scratch. Imagine a metabolically active respiring tissue cell. As it burns glucose, it constantly generates carbon dioxide as waste product. This creates a high partial pressure of carbon dioxide in the tissues compared to the systemic capillary. Because gas moves down its pressure gradient, carbon dioxide diffuses out of the tissue cells, crosses the interstitial fluid, passes through the capillary endothelial wall. This is the wall we're talking about, and enters the blood plasma. Once in the blood, this carbon dioxide must be transported to the lungs, and it utilizes three distinct pathways to get there. The first fraction of carbon dioxide simply dissolves directly in plasma. It doesn't undergo any chemical reaction or bind to any proteins. It just remains as dissolved gas traveling in the liquid portion of the blood. This is the dissolved portion of carbon dioxide. The remaining major fractions of carbon dioxide pass straight through the plasma and cross the erythrocytic membrane to enter the red blood cell. That means carbon dioxide was inside the tissue, it crossed the interstitium, it came to plasma, then it entered RBC. Once inside the RBC, the remaining portions, this and this, they enter inside red blood cell. Once inside red blood cell, one portion encounters hemoglobin molecules that just have delivered oxygen to the tissues. This carbon dioxide binds directly to the amino terminal groups of the globin proteins. Point to be noted, it doesn't bind to the heme iron, rather the globin protein of the hemoglobin molecule. And this binding forms this compound called carbaminohemoglobin. This is the largest fraction of carbon dioxide, and this portion undergoes a crucial biochemical sequence inside the cytoplasm of the red blood cell. The carbon dioxide combines with intracellular water. Under normal circumstances in the plasma, this hydration reaction is incredibly slow. However, inside the erythrocyte, it is instantly catalyzed by an abundance of the enzyme called carbonic anhydrase. Carbonic anhydrase rapidly converts carbon dioxide and water into carbonic acid. This is the carbonic acid, that is H2CO3. This carbonic acid is a weak and highly unstable acid. So it immediately dissociates into a hydrogen ion and a bicarbonate ion, HCO3-. This accumulation of products presents a challenge that the red cell must instantly resolve to maintain homeostasis. First, the rising concentration of this free hydrogen ion threatens to severely drop the intracellular pH. To buffer this acid, hydrogen ions bind to deoxygenated hemoglobin, written as Hb-. To buffer this acid, this proton first binds with the hemoglobin of this oxyhaemoglobin molecule, and as a result, it creates reduced hemoglobin. Here, H stands for the proton that we were getting previously from this reaction, and oxygen is released from the hemoglobin. Deoxyhemoglobin, that means hemoglobin that has released its oxygen, acts as an excellent buffer, and its affinity for protons actually stabilizes in its deoxygenated state, promoting further oxygen release to the tissues. Secondly, the bicarbonate ions accumulate inside the cell, creating a steep concentration gradient. That means the concentration of bicarbonate gets higher compared to the outside of RBC. And that moment, RBC uses a specialized membrane transport protein. Imagine this is the protein we were talking about. This is called band 3 protein. And this band 3 protein pumps out bicarbonate out of the erythrocyte and into the plasma, down its concentration gradient. However, pumping out a negatively charged ion creates an electrical imbalance. To maintain strict electrical neutrality across the membrane, a chloride ion is simultaneously pumped inside the red blood cell from the plasma in a one-to-one exchange. One-to-one means bicarbonate goes out and chloride goes in. This reciprocal movement of ions at the tissue level is what we call the chloride shift or hamburger shift. And now let's follow this blood as it returns to the right side of the heart and is pumped into the pulmonary capillaries, arriving at the lung alveoli. This is the border of alveolus, and this is the border of capillary, or this is the capillary endothelial wall, we can say. Here, the partial pressure gradient is completely reversed compared to the gas exchange at respiring tissues. The alveoli are filled with fresh atmospheric air. That means the partial pressure of oxygen is very high, so many oxygens here, and the partial pressure of carbon dioxide is very low, let's say 2, just to put into perspective. This high ambient oxygen level forces oxygen to diffuse from the alveoli into plasma. This oxygen is coming to plasma and finally into RBC. This is the oxygen we're tracking. This is just one example. This type of reaction keeps happening in millions of RBCs. Inside the RBC, it binds to hemoglobin to form oxyhaemoglobin. But hold on, when this oxygen forms oxyhaemoglobin, it first binds to this reduced hemoglobin, which has a proton attached to it. When this oxygen first binds to reduced hemoglobin, it causes a structural conformational change that drastically reduces its affinity for both carbon dioxide and hydrogen ions. And as a result, this proton or this hydrogen ion gets released back into the cytoplasm, shown here. This is the proton that got detached from the reduced hemoglobin we were talking. This sudden drop in intracellular bicarbonate and rise in free protons drives the entire chemical equilibrium backward. Bicarbonate's ions in the plasma are pulled back inside the red blood cell via the same band 3 protein, and to maintain electrical neutrality, chloride ions are pumped out of the cell back into the plasma. Once inside, the re-entered bicarbonate combines with the released proton to create carbonic acid. Carbonic anhydrase enzyme immediately catalyzes the reverse reaction, breaking this carbonic acid into water and carbon dioxide. Simultaneously, the carbaminohemoglobin releases its bound carbon dioxide, shown here as hemoglobin plus CO2. This massive wave of newly liberated gaseous carbon dioxide, along with fraction that was simply dissolved in plasma, shown here, diffuses rapidly across the alveolar capillary membrane. This is the capillary membrane and this is the alveolar membrane. These carbon dioxide diffuses into the alveolus. That means all of them come to the air spaces of the lungs where it is expired out of the body. Now look at the slide to reinforce the high yield facts for the exam. Starting off with how the carbon dioxide is transported. Approximately 70% of carbon dioxide is transported as bicarbonate, and it is produced inside the red blood cells and carried in the plasma. Carbaminohemoglobin represents the fraction of carbon dioxide bound directly to the globin protein chains of hemoglobin, accounting for approximately 20% of carbon dioxide transport. Dissolved carbon dioxide is the smallest transport fraction, accounting for roughly 7 to 10% of carbon dioxide, which travels simply dissolved as a gas within the blood plasma. And what is the tissue sequence of carbon dioxide transport? First, carbon dioxide enters RBC. Inside RBC, we have carbonic anhydrase, and it catalyzes to form carbonic acid. Carbonic acid is unstable and weak. That's why it rapidly breaks down into proton and bicarbonate. Bicarbonate exits the RBC and goes into plasma. And in exchange of that, chloride enters, which is known as chloride or hamburger shift. And the lung or alveolar sequence goes like, first, bicarbonate re-enters the RBC. It combines with proton to form carbonic acid. Carbonic acid is broken down into carbon dioxide and water by carbonic anhydrase. The released carbon dioxide is expired through the lungs. Now pay critical attention to these high-yield exam traps to protect your core. Examiners love to state that carbonic anhydrase is active in the blood plasma. This is absolutely false. Carbonic anhydrase is located inside red blood cells, not in plasma. Plasma carbon dioxide hydration without enzyme is too low to sustain life. Effectively, all chemical conversion of carbon dioxide to bicarbonate occurs strictly within the erythrocytes or RBCs. And do not confuse Haldane effect with another effect called Bohr effect. The Haldane effect states that deoxyhemoglobin has much higher affinity for binding carbon dioxide as carbaminohemoglobin. Therefore, at tissue level, the unloading of oxygen directly promotes the pickup of carbon dioxide. Conversely, at lung, the presence of high oxygen concentration, that means oxygenation, forces hemoglobin to release its bound carbon dioxide so it can be expired. Two examples. First one is the chloride shift. This process is mediated by a specific transport channel called band 3 protein that we have seen in the diagram. Its physiological purpose is to exchange intracellular bicarbonate for extracellular chloride to maintain electrical neutrality across the erythrocyte membrane during gas exchange. Then, Bohr effect and Haldane effect complementarity. The Bohr effect, which dictates that high carbon dioxide and acid levels drive oxygen delivery to tissue, and Haldane effect dictates that low oxygen levels drive carbon dioxide pickup. This Bohr effect and this Haldane effect are perfectly complementary processes. They work in tandem to maximize gas exchange efficiency in actively exercising skeletal muscle. Now let's map out the neural architecture. That means neural control governing the respiratory system completely from scratch. Unlike cardiac muscle, which possesses intrinsic myogenic automaticity, the muscles that drive breathing are entirely skeletal. Your diaphragm, intercostal, and accessory muscles require a continuous rhythmic neural drive from the central nervous system, CNS, contract. This rhythmic electrical command is generated and modulated within the brainstem, especially across the medulla and pons. Let's isolate the medulla first, as it functions as the primary rhythm generator. Deep inside the ventrolateral medulla lies a small cluster of interneurons known as Trebotzinger complex. Think of this microscopic cluster as the true rhythmogenic pacemaker of respiration. It spontaneously fires intrinsic burst of action potentials that establish the baseline respiratory rate, functioning analogously to the sinoatrial node of the heart. The rhythmic signals from this pacemaker project immediately to two primary functional aggregates of neurons within the medulla, the dorsal respiratory group and the ventral respiratory group. The dorsal respiratory group, or DRG, is located dorsally within the nucleus tractus solitarius. You don't need to remember all of them. I will tell you what you need to remember for the exam when we review the slide. But for a proper cohesive understanding, you need to know the full picture. The DRG, or dorsal respiratory group, is the principal engine that drives inspiration. When the pacemaker fires, neurons inside DRG light up and send excitatory somatic motor commands down the spinal cord. As you can see, the signal is being conveyed down the spinal cord with the help of nerve fibers. The signals that have been generated, it travels via the phrenic nerve. This is the phrenic nerve directly to the diaphragm, the main muscle of respiration, to diaphragm. And also it goes to the intercostal muscles that are in between the ribs. As these muscles contract, the thoracic volume expands, generating the negative intrathoracic pressure required for quiet, resting inspiration. In contrast, the ventral respiratory group, or VRG, is located in the ventrolateral medulla within the nucleus ambiguous and nucleus retroambiguous. The VRG contains a dual population of both inspiratory and expiratory neurons. During normal, quiet hypneic breathing, expiration is a purely passive process driven by the elastic recoil of the lungs, meaning the VRG is mostly quiescent. However, during active or forced breathing, such as intense exercise or respiratory distress, this VRG is recruited. Its inspiratory neurons reinforce the DRG, while its expiratory neurons fire to drive the internal intercostal muscles and the accessory respiratory muscles of the abdominal wall and chest, actively forcing air out of the lungs. Now let's move superiorly up to the brainstem into the pons. We will talk about the central nervous system and the structure of brain in the nervous system lecture. So don't get worried about it right now. Just follow along. Okay, so as we were talking about pons, this brain structure does not generate a respiratory rhythm. Instead, it acts as a group of modulators that fine-tune the switch off point and transition between inspiration and expiration. Within the pons, we identify two distinct antagonistic centers, the pneumotaxic center and apneustic center. The pneumotaxic center, also designated as the pontine respiratory group, resides in the upper pons. Its primary physiological job is to limit the duration of inspiration. It sends a continuous stream of inhibitory signals down to the medullary dorsal respiratory group. Its primary physiological job is to limit the duration of inspiration. It sends a continuous stream of inhibitory signals down to the medullary dorsal respiratory group, as you can see with the green colored curved arrow. I mean this one. By shutting off the inspiratory signal early, it limits the tidal volume and effectively regulates the rate of breathing. And positioned inferiorly to the pneumotaxic center, we see apneustic center. The apneustic center does the exact opposite. It sends excitatory signals to the DRG. That's why I written plus over here and the previous one was minus. So apneustic center sends excitatory signal down the DRG to prolong inspiration, preventing the inspiratory cutoff and encouraging deep gasping breaths. Under normal physiologic condition, the powerful pneumotaxic center, the first one, constantly overrides and balances the apneustic center, ensuring a smooth, coordinated transition into expiration. Now let's solidify what we have studied with the demonstration of the image. For exam, these informations are more than enough, but we study the diagram to have a clear picture so that you don't have to memorize, rather you can conceptually understand and keep this mechanism in your heart, because that's how you need to read biology. Okay, so first, we will talk about the respiratory rhythm generation. The baseline rhythm is generated by brainstem. Because respiratory muscles are skeletal, they require continuous, uninterrupted neural impulses from these centers to function, the medulla. This is the primary rhythm generator. First, we will talk about the pre-Botzinger complex. This functions as the true intrinsic rhythmogenic pacemaker of respiration. Then DRG, or dorsal respiratory group, it is located within the nucleus tactus solitarius. Primary drives inspiration by sending motor signals via the phrenic nerve to the diaphragm and intercostal muscles. Central respiratory group, it is located within the nucleus ambiguous and retroambiguous, contains both inspiratory and expiratory neurons to drive active forced respiration via internal intercostal and accessory muscles. Pons work as the modulators. Pontine respiratory group or pneumotaxic center limits the duration of inspiration, thereby controlling breathing rate and preventing overinflation. Apneustic center, it functions to prolong inspiration. Its activity is normally held in check and balanced by pontine respiratory group. Now you need to pay critical attention to these high-yield exam traps to protect your score, just like before. Medical boards frequently try to trick students by stating that pneumotaxic center or pontine respiratory group is located within the medulla. Always remember that pneumotaxic center is in the pons, not in medulla. Do not confuse the roles of the two brainstem regions. The medulla is the primary rhythm generator, whereas the pons acts purely as a modulator. If an exam question describes a lesion or traumatic damage to the pontine respiratory group, the inhibitory check on inspiration is lost. This allows the uninhibited apneustic center to dominate, resulting in apneosis, characterized by abnormal, prolonged inspiratory gasp. A classic experimental trap asks what happens if the entire pons is surgically removed or transected while leaving the medulla intact, because the medulla contains the primary rhythm generator and the true pacemaker. The medulla alone sustains regular breathing, even if the modulating pons is removed. The pre-Botzinger complex is so robustly hardwired as an essential pacemaker that it remains highly active even when completely isolated in neonatal tissue preparation, proving its standalone atomicity. It means that even if it is removed from the body, it can generate automatic rhythm, which we also would see in case of pacemaker of heart. And in severe head injuries or herniation syndrome, respiratory arrest occurs only with medullary center damage, not pontine. A pontine lesion will alter the pattern of breathing, but a catastrophic medullary lesion destroys the primary pacemaker drive, completely halting spontaneous respiration. Beyond the central, now let's sketch out the chemical control of breathing from first principles. Your body continuously monitors the chemical composition of your blood and cerebrospinal fluid, which is found in the ventricles of the brain, to ensure that ventilation perfectly matches metabolic demand. To achieve this, the respiratory system relies on two anatomically and functionally distinct population of sensors. One is central chemoreceptors, another one is peripheral chemoreceptors. Let's first draw the central chemoreceptors. The central chemoreceptors are located inside the central nervous system on the ventrolateral surface of medulla oblongata. These receptors are bathed in cerebrospinal fluid, or CSF, and are separated from the systemic circulation by the blood-brain barrier. Blood-brain barrier means a barrier in the brain that doesn't allow every component of blood to enter inside brain. Now imagine an increase in metabolic activity that raises the partial pressure of carbon dioxide in arterial blood. The blood-brain barrier is highly selective, as we were saying. It is a lipid bilayer, meaning it is impermeable to charged ions like hydrogen or bicarbonate. However, carbon dioxide is a gas and is highly lipid soluble, so it diffuses freely and rapidly crosses the blood-brain barrier from the capillaries into the CSF of the brain. Once inside the CSF, carbon dioxide encounters water and undergoes the familiar hydration reaction to form carbonic acid, which immediately dissociates into hydrogen ion and bicarbonate ion. Because the CSF has a very low concentration of proteins compared to plasma, its buffering capacity is incredibly poor. Consequently, the newly generated hydrogen ions cause a sharp drop in CSF pH. It is the local drop in pH, specifically the accumulation of hydrogen ions inside the CSF, that directly stimulates the central chemoreceptors. They signal the nearby medullary rhythm generators to instantly ramp up ventilation, blowing off excess carbon dioxide until the system returns to baseline. This was the whole story about central chemoreceptor. Now let's sketch the peripheral chemoreceptors, which are located entirely outside the brainstem to sample the systemic arterial blood before it reaches the rest of the body. The sensors are organized into two major structures, the carotid bodies and the aortic bodies, indicated with these dots colored in green. Let's look at the bifurcation. That means this carotid artery has bifurcated into two new branches. In this bifurcation, tucked inside the carotid sinus are the carotid bodies. When they detect chemical alteration in the blood, they transmit their sensory information up to the brainstem via the glossopharyngeal nerve. These green colored wires are the nerves, and specifically this nerve is the glossopharyngeal nerve or cranial nerve number 9. One you see on the left side and one on the right. Down below, along the superior surface of the aortic arch are the aortic bodies. The sensory fibers originating from these structures travel back to the brainstem via the vagus nerve. One vagus nerve here, another one here, which is cranial nerve 10. Down below, unlike the central chemoreceptors, which are completely blind to oxygen levels, the peripheral chemoreceptors are your body's primary oxygen sensors. They respond to three distinct arterial stressors. First, a drop in arterial oxygen tension or drop in partial pressure of oxygen. Number 2, rise in partial pressure of carbon dioxide inside the arteries. And 3, a drop in arterial blood pH. Under normal physiological conditions, these peripheral sensors maintain a baseline low-frequency firing rate. However, if the partial pressure of oxygen in the arterial blood plunges, these receptors dramatically accelerate their firing rate. They send rapid excitatory signals up cranial nerves 9, which is glossopharyngeal nerve, paired, and vagus nerve paired, and this with glossopharyngeal nerve and vagus nerve straight into medullary respiratory groups, triggering an immediate powerful hyperventilation to salvage systemic oxygenation. Hyperventilation means your breathing will increase, and if your breathing increases, the body gets rid of the excess carbon dioxide, which brings the pH of the artery to normal, and also it removes the excess carbon dioxide, and it also balances out the oxygen level. So we have covered the definition and key features both at the same times. You need to read them through properly, and if you want them put together in one place, you can check my biology living note. And watch out, don't fall prey to these exam traps if you want to protect your score. The first one is central oxygen sensor illusion. A classic board question will trick you into stating that central chemoreceptors respond to hypoxia. Hypoxia means low oxygen. But the fact is, central chemoreceptors have absolutely no sensitivity to oxygen. That's why it's written central receptors do not directly sense oxygen or arterial carbon dioxide. They're driven entirely by pH changes in the CSF. Examiners frequently state that systemic metabolic acidosis. That means the second one is blood-brain barrier ion trap. Examiners frequently state that systemic metabolic acidosis, that means high arterial hydrogen ion, immediately triggers central chemoreceptors. This is false because hydrogen ion and bicarbonate cannot cross the blood-brain barrier. Only arterial carbon dioxide can cross blood-brain barrier freely because carbon dioxide is lipid soluble. The third one is the mild hypoxia trap. Do not assume that peripheral chemoreceptors fire strongly the moment partial pressure of oxygen drops slightly below 100. This is not correct. Because of the plateau phase of the oxygen hemoglobin dissociation curve, hemoglobin saturation remains high even with moderate drop in oxygen pressure. Consequently, peripheral chemoreceptors only activate strongly when the partial pressure of oxygen falls below 60 mm of mercury. And we got a couple of examples. First one is COPD hypercapnia. In patients with severe chronic obstructive pulmonary disease or COPD, as written here, long-term trapped carbon dioxide causes hypercapnia. Over days, a specific part of brain pumps bicarbonate into CSF to neutralize the acid. This causes the central chemoreceptors to reset and lose their drive. Consequently, the patient's respiratory drive shifts entirely to the peripheral chemoreceptors, making carotid body the primary driver of breathing via hypoxia. The second peril is the danger of high flow oxygen. If you give a chronic COPD patient high flow oxygen, you abruptly raise their partial pressure of oxygen above 60 mm of mercury. This removes their hypoxic drive, leading to acute hypoventilation, severe respiratory acidosis, and it can cause unconsciousness. In clinical practice, you must use titrated oxygen to maintain a target oxygen saturation of 88 to 92% saturation of oxygen. Titrated oxygen means adjusting the level of oxygen based on the need of the patient. Now that we have thoroughly covered the previous anatomies of the central and peripheral chemoreceptors, we can fast forward to the topic of Hering-Breuer reflex and pulmonary receptors. These are mechanical sensors in the lung itself that feed information back to the brainstem. And out of them, the most famous is the Hering-Breuer reflex. In the year 1868, Hering and Breuer discovered that inflating the lung inhibited further inspiration. This is the Hering-Breuer inflation reflex, and it is mediated by slowly adapting stretch receptors in the smooth muscle for the airway. When the lung inflates sufficiently, inflating the lung inhibited further inspiration, and this is the Hering-Breuer reflex, and it is mediated by slowly adapting stretch receptors in the smooth muscle of the airway, like bronchus or trachea. When the lungs inflate sufficiently, these receptors fire as they detect the inflation, and the receptors send signals via the vagus nerve to the dorsal respiratory group of the medulla, where they inhibit the next respiratory burst. There are two other receptor types worth knowing: rapidly adapting or irritant receptors. They are located in the airway epithelium. They respond to inhaled irritants, smoke, cold air, chemicals, and mechanical distortion. When they fire, they trigger coughing, bronchoconstriction, that means the bronchus gets narrower, and hyperapnea. Hyperapnea is breathing deeper and faster than normal. If you ever have inhaled pepper and sneezed violently, you have experienced these receptors. J receptors, or juxtacapillary receptors, are found in the alveolar walls adjacent to the pulmonary capillaries. They are stimulated by the engorgement of the pulmonary vasculature, that means pulmonary vessels, and by accumulation of fluid in the alveolus, alveolar interstitium, as occurs in pulmonary edema. That means buildup of fluid inside the lungs. When J receptors fire, they produce the subjective sensation of breathlessness, dyspnea, and a drive pattern of rapid, shallow breathing. This is one reason patients with heart failure and pulmonary edema feel so breathless. So let's review the mechanism once again. Lung will inflate, and as a result, the stretch receptors in the airway will fire. The signal will go through the vagus nerve to the dorsal respiratory group of the medulla, and it will be inhibited. That means it will be stopped, and as a result, expiration will be triggered. It will be very easy to understand if you could follow along the previous slides that I taught in details. Many students mistakenly believe the Hering-Breuer reflex controls every normal breath. The reality depends heavily on age and physiological state. You need to remember the primary off switch to end inspiration is spontane respiratory group. This Hering-Breuer reflex stops inspiration via lung stretch receptors to prevent overinflation. That's why this is not a dominant breath terminator. In resting adults, only activates when the tidal volume exceeds 1 liter. You gotta know, normal resting tidal volume is only 500 ml. If you forget, then refer to the previous section. It is highly active and dominant in newborns and anesthetized animals. That means the animals who have been administered anesthesia. The first example talks about vagotomy, or cutting the vagus nerve. This will remove stretch receptor input, and the result would be inspiration is no longer inhibited early, and breathing pattern becomes slower and deeper. The J receptor activation we talked about, it is activated by fluid buildup such as edema, and it triggers rapid shallow breathing and severe dyspnea. Dyspnea means shortness of breath. Let us now pull all of these mechanisms together by asking what happens to a person who travels from sea level to high altitude. It gives birth to a concept of altitude acclimatization. And this scenario threads together the ODC, that means oxygen dissociation curve, chemoreceptors, kidney physiology, and red cell adaptation in one stone. IMAT loves integrated scenarios. First, the basic physics. Atmospheric pressure decreases with altitude. At the sea level, the pressure is 760 millimeters. At 5,000 meters, the summit of many Himalayan base camps, it is approximately 380 millimeters. But the fractional composition of the atmosphere doesn't change. Oxygen is still 21% of the atmosphere at every altitude. So alveolar PO2, or alveolar partial pressure of oxygen at 5,000 meters, is roughly 50 millimeters of mercury instead of 100. Now let's come to the mechanism or sequence. The first phase of the altitude acclimatization is the acute phase or acute response, which is minutes to hours. In acute phase, partial pressure of oxygen falls below 60 millimeters of mercury. The carotid bodies, which work as peripheral chemoreceptors, fire. They drive hyperventilation. That means ventilation increases over normal level, and carbon dioxide is blown off. As a result, the partial pressure of carbon dioxide falls. We know carbon dioxide is acidic. When the carbon dioxide partial pressure drops, it creates respiratory alkalosis. This alkalosis causes a left shift of oxygen dissociation curve, increasing hemoglobin's oxygen affinity. That means hemoglobin will like to have more oxygen than in normal case. Paradoxically, this temporarily impairs oxygen delivery to tissues, even as ventilation increases. That's why it is written transiently impairs tissue oxygen delivery, which is paradoxical in nature. This acute maladaptive shift is quickly overridden by acclimatization, and this process kicks in over days to weeks. The kidneys detect the alkalosis and compensate by excreting bicarbonate ion. This lowers the plasma bicarbonate, normalizes pH, and compensates for alkalosis, which allows the ventilatory drive to continue, written here as permit further hyperventilation. And in response to chronic hypoxia, that means deficiency of oxygen, RBC increase their production of 2,3-BPG. 2,3-BPG binds to deoxyhemoglobin and stabilizes the T state, causing a right shift in ODC. This right shift increases oxygen delivery to tissues. And over weeks, the kidneys release erythropoietin, EPO. EPO stimulates bone marrow to produce more red blood cell, and hematocrit and hemoglobin concentration rise as well. Total oxygen carrying capacity of the blood increases. At full acclimatization, the individual can sustain reasonable activity at altitude, because the combination of hyperventilation, right-shifted ODC, and increased hemoglobin concentration compensates for the low inspired partial pressure of oxygen. These are the traps that you shouldn't fall for. These are the traps that you shouldn't fall prey to. First one, to review the whole concept, let's look at the traps and examples once again. Remember, when you first ascend, hyperventilation causes respiratory alkalosis. This gives you an acute left shift in oxygen dissociation curve. That means it makes hemoglobin hold onto oxygen too tightly. But give it a few days and your body acclimatizes. It increases 2-3 bpg and causes a right shift to improve tissue delivery. Don't confuse this with EPO. 2-3 bpg takes days while EPO takes weeks. Now for our first example, acetazolamide. If a patient has mountain sickness, this carbonic anhydrase inhibitor is the drug of choice. It forces the kidneys to dump bicarbonate that directly cancels out the altitude-induced alkalosis, rapidly speeding up renal compensation. Our second example is hypoxic pulmonary vasoconstriction. Remember, systemic vessels dilate to hypoxia, but primary vessels constrict. Remember, systemic vessels, that means aorta, superior vena cava, inferior vena cava, the vessels that are connected to the systems of our body, they dilate to hypoxia, but pulmonary vessels constrict. By constricting blood vessels in poorly oxygenated parts of the lung, the body shifts blood toward well-ventilated alveoli to maximize oxygen pickup. That's why it's written that in hypoxia, pulmonary vasoconstriction occurs, and as a result, the blood is redirected from poorly ventilated alveoli to the well-ventilated alveoli. Let's break down obstructive pathology from first principles by comparing two major clinical entities, asthma and COPD. While both conditions cause expiratory airflow limitation and share an obstructive pattern on spirometry, their underlying cellular mechanics, tissue remodeling, are radically different. Let's look at asthma first. In asthma, the airway is highly inflammatory, and when the bronchioles, the airways, are exposed to an environmental trigger, this bronchioles forcefully contract and narrows the lumen. Simultaneously, airways swell due to edema, and the narrowed airway gets filled with thick mucus, and this creates massive resistance to airflow during expiration. But interestingly, the smooth muscle of the asthma patient eventually relaxes, meaning asthma is fundamentally a reversible disease. Now let's contrast this with COPD, or chronic obstructive pulmonary disorder. This is a reversible progressive disease, and it has two phenotypes. One is bronchitis, another one is emphysema. Now let us sketch the chronic bronchitis first. Imagine an individual exposed to decades of irritating cigarette smoke. This chronic irritation causes persistent low-grade inflammation of the central and peripheral airways. The hallmark pathology here is mucus hypersecretion. This mucus hypersecretion clogs the bronchiole. Seen here inside the bronchiole, you see it is being clogged with mucus. And also, the walls of alveoli are destroyed here, which forms larger alveoli instead of normal or smaller ones. And this causes the drop in gas exchange. Hypoxemia, that means blood faces deficiency of oxygen, and cyanosis as well, which is bluish discoloration of the skin, as carbon dioxide is heavily retained. The cyanotic fluid retentive clinical presentation is classically termed as blue bloater. Now let's sketch emphysema. Actually, this portion describes the condition of emphysema. The primary pathology here occurs at the distal air sacs, that means the alveoli. Chronic cigarette smoke disrupts the delicate balance within the lung tissue. As a result, the elastic alveolar walls are destroyed, which creates larger floppy alveoli or air sacs. This anatomical destruction causes massive loss of surface area for gas exchange. During expiration, these larger air sacs collapse early, trapping immense volume of air inside the chest. To compensate for this structural collapse and loss of surface area, patients must relentlessly hyperventilate By keeping their respiratory rate incredibly high, they manage to maintain normal arterial oxygen and carbon dioxide level, but they face muscle wasting and weight loss. This thin hyperventilating presentation is classically termed as pink puffer. Now let's come to the famous exam traps. The first one is the blue bloater pathological tetrad. When an exam vignette describes a blue bloater, do not just look for chronic bronchitis and mucus hypersecretion. You must instantly link this phenotype to chronic hypoxemia, which triggers reactive polycythemia, that means excess RBC production, which increases the risk of pulmonary hypertension and cor pulmonale, which is the heart failure, the right-sided heart failure. Then the pink puffer, blood gas illusion. Do not assume all severe COPD cases have high carbon dioxide levels. A classic pink puffer emphysema patient utilizes an incredibly high respiratory rate to maintain near normal carbon dioxide level. They will appear thin, breathless, and barrel-chested, but their blood gases are often deceptively close to normal, which is a hallmark of emphysema. Examiners love to test your understanding of lung volumes in emphysema. Because of the loss of alveolar walls, the lungs become highly compliant and easily trap air. This causes a dramatic increase in total lung capacity and residual volume. Never mistake a high TLC for a restrictive pattern. In emphysema, a high TLC is a hallmark sign of permanent air trapping. Now the exam pearls. First one is alpha-1 antitrypsin deficiency. While cigarette smoke is the primary cause of COPD by disrupting the protease and antiprotease balance, always screen for alpha-1 antitrypsin deficiency if an exam vignette describes a young non-smoker presenting with early onset emphysema. The keyword here is non-smoker. The uniform obstructive metric. We will study about that in this primary cause of COPD. Regardless of whether the patient presents with emphysema, chronic bronchitis, or asthma, all of them are obstructive lung diseases. Therefore, they will all share a hallmark diagnostic spirometry finding. And that finding is the FEV1 versus FEV1 and FVC ratio will be below 0.7. Now let us close this lecture with a high-speed walkthrough of every fact that is most likely to appear on your IMAT paper. I want you to be able to fire this out without hesitation. Let's review the high-yield anatomical structural landmarks of the respiratory tree. If you look at the bifurcation of the trachea, the right primary bronchus is physically shorter, significantly wider, and runs much more vertically compared to the left bronchus. Because of this steep vertical pathway, inhaled foreign bodies or aspirated fluids have a strong anatomical preference to enter and lodge inside the right lung, which is known as aspiration, and it is written as aspiration preference. That means right bronchus is more prone to aspiration risk. Deep within the alveolar walls, we find type 2 pneumocytes. These specialized epithelial cells act as microscopic manufacturing plants of surfactant, and at the same time, type 2 pneumocytes also serve as progenitor cells that proliferate and regenerate injured type 1 pneumocytes to maintain the integrity of the blood-air barrier. When evaluating the mechanism of ventilation, quiet normal inspiration is always an active process, requiring skeletal muscle contraction. In contrast, quiet expiration is entirely passive, driven completely by the elastic recoil of the lung tissue and chest wall. Looking at clinical diagnostic metrics, three specific lung volumes cannot be directly measured by standard static spirometry. They are RV, residual volume, functional residual capacity, and total lung capacity. Because FRC and TLC both mathematically include residual volume, you must use specialized technique like helium dilution to calculate them. That's why it's written not measurable by spirometry. Let's clear up a common baseline metric trap regarding gas pressures. The partial pressure of oxygen inside our alveoli 100 millimeter mercury, not 159 found in dry atmospheric air. The significant drop occurs because as inspired air travels down the conducting zone to reach body temperature at 37 degrees centigrade, it becomes fully saturated with water vapor, which exerts its own pressure and dilutes the oxygen molecule. When looking at gas diffusion kinetics across the alveolar capillary membrane, carbon dioxide diffuses 20 times faster than oxygen gas. This massive speed diffusion is driven entirely by the high solubility coefficient of carbon dioxide. Due to this reason, hypoxia precedes hypercapnia in disease forms. For systemic circulation transit, carbon dioxide transport uses three discrete pathways. Approximately 70% travels as bicarbonate ions in the plasma, roughly 20% travels as carbaminohemoglobin bound directly to the globin chains of hemoglobin, and the remaining 7 to 10% travels simply dissolved as a gas within the plasma. The biochemical process relies entirely on the fact that enzyme carbonic anhydrase is located strictly inside the cytoplasm of red blood cells, completely absent from the extracellular plasma. The resulting accumulation of bicarbonate forces an exchange through the cell membrane, where the chloride shift or Hamburger shift moves a chloride ion inside to maintain perfect electrical neutrality. To master oxygen hemoglobin dissociation curve, remember the memory acronym CADET PACE, right? The letters stand for C, carbon dioxide, A, acid, 2,3-BPG, E for exercise, T for temperature. An increase in any of these factors shifts the curve to the right. A rightward shift means oxygen's affinity for oxygen, hemoglobin's affinity for oxygen drops, resulting in higher P50 value, and it directly facilitates better, more efficient oxygen delivery to respiring tissues. Conversely, toxic gases can disrupt this binding curve. Carbon monoxide binds to heme iron of hemoglobin with an affinity of 250 times more than oxygen, and it causes left shift. We see a physiological high affinity state naturally in fetal hemoglobin, or HbF, structurally composed of 2 alpha and 2 gamma globin chains. This hemoglobin F exhibits a distinctly left-shifted dissociation curve, and this elevated affinity allows the fetus to extract oxygen from maternal hemoglobin A. Looking at the neural control loop, central chemoreceptors are clustered on the ventrolateral surface of medulla oblongata. They are completely blind to oxygen levels, responding instead to drops in cerebrospinal fluid pH, driven directly by arterial carbon dioxide crossing the blood-brain barrier. Meanwhile, peripheral chemoreceptors are situated outside the brain in the carotid bodies and aortic bodies, transmitting signals via the glossopharyngeal nerve, which is cranial nerve number 9. This functions as your primary oxygen sensor, but they keep a quiet baseline and only accelerate their firing rate dramatically when arterial oxygen pressure drops sharply below 60 mm of mercury. This incoming sensory input is integrated by the pneumotaxic center, or pontine respiratory group, located up in the pons. The pneumotaxic center functions as the primary, indispensable neural off switch for inspiration in adults, limiting the tidal volume to regulate a smooth respiratory rate. In severe chronic respiratory conditions like COPD, the resulting hypercapnia, that means excess carbon dioxide, causes the central chemoreceptors to completely reset. The patient's entire respiratory drive shifts to a hypoxic drive driven by the carotid bodies. If a clinician administers high-flow oxygen, they shut down this remaining hypoxic sensor, introducing catastrophic risk of severe hypoventilation. Finally, let's look at environmental pressure changes. At high altitude, acute exposure causes compensatory hyperventilation that blows off massive amounts of carbon dioxide. The resulting respiratory alkalosis forces an acute leftward shift of the oxygen dissociation curve. However, over days of full acclimatization, the kidneys excrete bicarbonate and the erythrocyte upregulates 2,3-BPG production, shifting the curve back to the right to optimize tissue oxygen unloading. Alright, now we have come to the final segment of our lecture where we will solve IMAT style questions to bolster our understanding. The first question reads, which of the following lung volumes or capacities cannot be measured by spirometry alone? And we are given 5 options. To find the right answers, let's look at how spirometry actually works. A spirometer measures the volume of air an individual can inhale or exhale as a function of time. This means it's incredibly efficient at measuring dynamic air movement. Because of this, it easily tracks your tidal volume, the normal air breathed in and out at rest, as well as your inspiratory and expiratory reserve volume, which are the extra amounts you can force in or out. If we can measure these individual reserve volumes, we can also calculate vital capacity, which is simply the total amount of air you can fully exhale after taking your deepest breath. Since spirometry directly captures all of this, options A, B, C, and E are completely measurable and can be ruled out. That leaves us with option D, functional residual capacity. FRC is defined as the volume of air remaining in the lungs after a normal passive exhalation. Why can't spirometry measure this? Because FRC includes the residual volume, the trapped air that physically cannot be forced out of the lungs no matter how hard you try. Since a spirometer only records moving air, it cannot measure static trapped air. Therefore, FRC cannot be determined by spirometry alone. To find the functional residual capacity or residual volume in a clinical setting, we must rely on advanced techniques like helium dilution, nitrogen washout, or body plethysmography. If a capacity contains residual volume, spirometry alone won't cut it. Moving straight into question number 2, we encounter a crucial clinical scenario that builds perfectly on our understanding of respiratory control. The prompt describes a patient with severe COPD and chronic carbon dioxide retention who is given high flow oxygen. Suddenly, his respiratory rate falls dramatically and his partial pressure of carbon dioxide rises even further. And the question is, which of the following best explains this? Let's break down exactly why this happens. In a healthy individual, the primary stimulus to breathe comes from central chemoreceptors reacting to elevated carbon dioxide. However, in a patient with chronic COPD, their carbon dioxide levels are persistently high. Over time, the brain adjusts and becomes completely desensitized to this hypercapnia. As a result, these patients lose their primary carbon dioxide drive. They become entirely dependent on peripheral chemoreceptors, which monitor low oxygen levels instead. This baseline state is known as hypoxic drive. When this patient is suddenly given high flow oxygen, the baseline hypoxia is rapidly corrected. Because the peripheral chemoreceptors sense that oxygen levels are now abundant, the brain concludes it no longer needs to work as hard. It essentially turns off the backup hypoxic drive. With the stimulus removed, the breathing rate plummets, trapping even more carbon dioxide. This directly points us to option C, the removal of hypoxic drive to breathe in a carbon dioxide tolerant patient. Now, a bit of nuance and alternative options. While modern physiology also points to factors like the Haldane effect and altered ventilation perfusion matching, which are vaguely related to option E, board exams traditionally focus heavily on the disruption of hypoxic drive. The alternative choices like oxygen toxicity or absorption atelectasis are real medical phenomena but wouldn't cause such an acute immediate crash in the patient's respiratory rate. So C is our final answer to 92%. So this clinical pearl is exactly why we target a lower oxygen saturation of 88 to 92% in severe COPD patients. It keeps them safe without turning off their internal clock to breathe. Now that we've mastered this concept, let's keep the momentum going and look at our next question. Question 3 shifts our focus from respiratory regulation to gas transport in the tissues. The question asks, what shift of oxyhemoglobin dissociation curve would you expect in vigorously exercising skeletal muscle? Let's look at the options and figure out how hemoglobin behaves under stress. To solve this, we just need to think about what is actually happening inside a muscle that is working incredibly hard. Vigorously exercising skeletal muscle has a massive demand for energy. Let's say this is the skeletal muscle. As a byproduct of this rapid metabolism, the muscle produces large amounts of carbon dioxide. It also undergoes anaerobic metabolism, generating lactic acid, which drops the local tissue pH. Additionally, all of this cellular work generates quite a bit of metabolic heat, raising the temperature. Now, how does hemoglobin respond to this hot, acidic, and carbon dioxide-rich environment? It responds by loosening its grip on oxygen. That means hemoglobin reduces its affinity of oxygen. This is a brilliant physiological design known as the Bohr effect. High carbon dioxide, low pH, and high temperature all decrease hemoglobin's affinity for oxygen, shifting the entire oxyhemoglobin dissociation curve to the right. A right shift means that at any given oxygen pressure, hemoglobin is much more willing to unload and dump oxygen directly into the tissues that need it most. Looking back at our choices, option C perfectly matches this physiology. A right shift due to increased carbon dioxide and decreased pH. We can easily rule out option A and B because left shifts mean hemoglobin holds onto oxygen tighter, which would starve a working muscle. Option D mentions right shift, but attributes it to a decrease in temperature when exercise actually increases it. Option E is false because this curve is highly dynamic rather than fixed. So remember the simple mnemonic: caret, face, right. C for carbon dioxide, A for acid, D for DPG or 2,3-DPG, E for exercise, T for temperature. This all shift the curve to the right, ensuring oxygen goes exactly where the metabolic fire is burning. Let's carry this concept forward as we look at our next problem. Moving forward to question number 4, we stay with the theme of gas transport, but we are flipping the script from oxygen over to carbon dioxide. The question asks, how is the majority of carbon dioxide transported in venous blood? Let's look at the options and break down the 3 primary ways our body handles carbon dioxide waste. When tissues produce carbon dioxide, it diffuses into the bloodstream, where it is carried back to the lungs in 3 distinct forms. First, a small fraction, only about 7%, remains completely unchanged and travels simply dissolved in plasma, which matches option A. Second, roughly 20 to 23% enters the red blood cells and binds directly to amino acid chains on hemoglobin molecules, forming carbaminohemoglobin as described in option B. That leaves the massive remaining balance, nearly 70% of all carbon dioxide. To handle this majority, carbon dioxide enters the red blood cell and undergoes a rapid chemical transformation catalyzed by the enzyme carbonic anhydrase. It combines with water to form carbonic acid, which immediately dissociates into hydrogen ion and bicarbonate ions. These bicarbonate ions are then pumped out into the plasma in exchange for chloride. Because roughly 70% travels this way, option C, as bicarbonate ions in plasma, is our correct answer. It is easy to get tripped by option D, carbonic acid inside the red blood cell. But remember that carbonic acid is highly unstable and transient. It doesn't stay in that form. Option E, binding to albumin, plays a negligible role in transport compared to hemoglobin. Therefore, bicarbonate in the plasma reigns supreme as the primary vehicle. Advancing naturally to question 5. We step away from gas transport dynamics and look directly at the microscopic structure of the lungs. The question asks, surfactant is produced by which type of alveolar cell? Let's review the main players lining our alveoli so we can confidently pick the right factory. This is alveolus, and the alveolar wall is primarily composed of two distinct epithelial cell types, type 1 and type 2 pneumocytes. Type 1 pneumocytes are incredibly thin. Let's say this is type 1 pneumocyte, T1. They cover about 95% of the alveolar surface area like this. 95% is covered by them and are optimized purely for gas exchange due to their minimal thickness. Because they are structural rather than secretory, we can rule out option A. That brings us to type 2 pneumocytes. These are plump cuboidal cells like this that account for the remaining surface area. Type 2 pneumocytes have two vital jobs. First, they act as stem cells that proliferate and replace damaged type 1 cells. That means it can make type 1 cells when needed. Second, and most importantly for this question, they synthesize, secrete, and recycle pulmonary surfactant. Surfactant is a complex mixture of phospholipids and proteins that reduces surface tension inside the alveoli, preventing them from collapsing during expiration. Therefore, option B, type 2 pneumocytes, is our correct answer. Now let's clean up the distractor. To keep our anatomical map clear, let's briefly dismiss the other options. Alveolar macrophages, our dust cells, are immune system's cleaning crew that clears out debris. Clara cells, now widely known as club cells, do secrete a surface active agent, but they reside in the bronchiole, not the alveoli. Finally, goblet cells secrete mucus and are found much higher up in the conducting zone, not down in the gas exchanging unit. A classic clinical tie-in to remember here is the neonatal respiratory distress syndrome, where premature infants lack mature type 2 pneumocytes, leading to a severe shortage of surfactant and widespread alveolar collapse. With this cellular puzzle solved, let's keep pressing forward to our next question. Moving seamlessly into question number 6. Now we'll leave the cellular structure of the lungs behind and scale new heights, literally. The question asks, at high altitude, which of the following is an adaptation that improves oxygen delivery to tissues? Let's examine how the human body masterfully copes with a low oxygen environment. When you travel to a high altitude, let's say over a mountain, the atmospheric pressure drops, meaning there are fewer oxygen molecules available with every breath. Your body immediately notices this hypoxemia and activates peripheral chemoreceptors. This causes you to increase your ventilation, not decrease it, ruling out option A because it said decreased ventilation, which is the opposite. This rapid breathing blows off carbon dioxide, the hyperventilation I mean. As carbon dioxide gets out from the body, it causes respiratory alkalosis, completely eliminating option D, as it is said acidosis here. To counter the effects of alkalosis and help release oxygen to starving tissues, red cells rapidly increase their production of metabolic byproduct called 2,3-BPG. As we learned back in question 3, an increase in 2,3-BPG binds to hemoglobin and decreases its affinity for oxygen, effectively shifting the oxyhaemoglobin dissociation curve to the right. That means if the curve was here, now it will get shifted to right. This right shift makes it much easier for hemoglobin to offload its precious cargo into oxygen-deprived tissues. Therefore, option C is our correct answer. Let's quickly clarify why the remaining options are incorrect. Over days to weeks, your kidneys release erythropoietin to increase hemoglobin synthesis, not decrease it, as option B suggests, so you can carry more total oxygen. Additionally, your body increases cardiac output in the short form to keep blood moving faster, eliminating option E. Now moving to question 7, which of the following statements about the central chemoreceptors is correct? This is a done deal question. We have already talked about it several times. Dismissing all wrong options, we will straight talk about the answer. We said central chemoreceptors are blind to bicarbonate or hydrogen ion levels. They respond to changes in CSF pH driven by carbon dioxide diffusion across the blood-brain barrier. So C is our correct answer. Advancing to question number 8. we put our clinical skills to the test with data interpretation. The question states that a patient has FEV1 of 1.8 liter and FVC of 3.6 liter. We need to find the most consistent interpretation among our choices. Let's run the math to see what these numbers reveal. First step is calculating the key ratio. Whenever you're handed spirometry data, your very first step should always be to calculate FEV1 to FVC ratio. The ratio tells us that percentage of their total vital capacity patient can forcefully exhale in very first second. Here, if we divide 1.8 liters by 3.6, we get exactly 0.5 or 50%. In a healthy adult, a normal FEV to FBC ratio is typically between 70 to 80%. Because this patient's ratio is severely dropped at around 50%, we know there is a clear expiratory airflow limitation. This hallmark reduction in the ratio is the defining characteristic of an airway obstruction, commonly seen in conditions like asthma or COPD. Since we don't have the patient's predicted baseline values, diagnose a true restriction, this clear drop in the ratio points us directly to option B, obstructive pattern only. Let's see why the other options don't fit. Option A is out because a 50% ratio is highly abnormal and it says normal here, so it's out. Option C is wrong because a pure restrictive pattern like pulmonary fibrosis typically preserves or even increases the ratio above 80%. Option D implies a mixed pattern, but we can't confirm a restrictive component without total lung capacity measurement. Finally, option E is a distractor. While a flow volume loop helpful, these absolute numbers give us more than enough data to find the obstructive defect. Moving naturally into question number 9, we encounter a classic toxicology topic with major physiological consequences. The question asks why carbon monoxide poisoning is so dangerous. Let's look closely at how this colorless, odorless gas attacks our oxygen delivery system. Carbon monoxide is incredibly dangerous because it has a massive competitive advantage over oxygen. It binds to the exact same iron sites on the hemoglobin. Let's say this is the hemoglobin. Carbon monoxide binds exactly to the place where oxygen were supposed to bind. However, its binding affinity is roughly 250 times more compared to oxygen. This means even tiny concentrations of carbon monoxide can rapidly crowd out oxygen and take over the transport vehicle, that is, hemoglobin. But the danger doesn't stop at simple competition. When carbon monoxide binds to one of the 4 sites on hemoglobin molecule, 1, 2, 3, 4, let's say carbon monoxide binds in this site, it physically alters the shape of the remaining subunit. The structural change increases the grip of the remaining sites on whatever oxygen is already bound. This locks the oxygen in place. They are almost locked in here, which causes a severe left shift of the ODC or oxygen hemoglobin dissociation curve. Because the curve shifts left, hemoglobin refuses to unload its oxygen into starving tissues. Therefore, option C is our correct answer, which says that carbon monoxide binds hemoglobin with much higher affinity than O2 and causes a left shift of the ODC for remaining oxyhemoglobin. Let's see why the other choices don't fit the pathophysiology. Carbon monoxide does not destroy type 2 pneumocytes, nor does it decrease surfactant, ruling out option A. It doesn't block carbonic anhydrase, which eliminates option B. It causes tissue hypoxia leading to lactic acidosis, not metabolic alkalosis, mentioned in option D. Finally, it doesn't physically alter alveolar surface area, eliminating option E as well. Now we come to our final question of this question solving session. And this question shifts our focus to the neural control network that regulates our automatic breathing rhythm. Question 10. It asks, during quiet breathing, what triggers the transition from inspiration to expiration? Let's take a look at the neural centers that dictate when a breath ends. Normal quiet breathing is a rhythmic process driven automatically by brainstem. The initial signal to inhale comes from the dorsal respiratory group or DRG located in the medulla. The DRG sends out a steady electrical signal that tells our primary inspiratory muscles like diaphragm to contract. However, to prevent our lungs from overinflating, this inspiratory RAM signal must be regularly turned off so passive expiration can begin. This critical switch-off mechanism is regulated higher up in the brainstem by the pontine respiratory group, or PRG, which includes the pneumotaxic center. This PRG acts like an internal timer. After a set period of inspiration, it sends inhibitory signals down to medulla to cut off the inspiratory signal. This clean cutoff allows the inspiratory muscles to relax, triggering a smooth transition into a passive exhalation. Therefore, option C is our correct answer, that reads pontine respiratory group inhibiting respiratory drive after a set time. While other options don't fit quiet breathing control. Chemical factors like rising carbon dioxide or falling oxygen levels, option A and option B, regulate the overall rate and depth of breathing over time, but they don't trigger the immediate breath-by-breath switch. J receptors in option D are sensory receptors in the alveolar walls that respond to injury or pulmonary edema. Finally, option E is incorrect because quiet expiration is entirely passive, meaning the internal intercostal muscles do not contract. And that brings us to the end of this deep dive masterclass on the respiratory system here at IMAT Tutor. I know this has been an incredibly long, comprehensive lecture, but it is packed with absolute utility for your preparation. We have methodically built every single concept from first principle, from the biophysics of gas transport and the chloride shift, all the way to the neural loop of brainstem and complex clinical pathophysiology. I can confidently guarantee you that everything you will ever encounter on the IMAT regarding the respiratory system was covered in this video. There are no stones left unturned, and no examiner tricks that can catch you off guard if you have mastered these anchor slides, exam traps, and pearls. If you found this masterclass valuable, make sure to hit that subscribe button, give this video a thumbs up, and share it with your fellow medical applicants. Keep studying hard, stay disciplined, and I will see you all in the next high-yield IMAT Tutor lecture.